So actually it's a great pleasure to be here and to have the opportunity to share with you some of our ideas, concepts, and some results concerning mislocalization of proteins in human diseases and the development of therapeutic strategies. But first, let me begin with some very basic things. So life or biological systems evolved to perform chemistry. Chemistry in water and almost exclusively between 0 and 100 degrees. So when the chemistry becomes more complex in eukaryotic cells, reaction compartments evolved, so-called organelles. So there are a lot of organelles around, different organelles with different metabolic activities and all with different proteins. But the production, the synthesis of proteins is confined just in one single compartment. So the translocation of proteins is a fundamental requirement of cell physiology. And in fact, more than half of the proteins have to be transported into or across biomembranes. So, as you can imagine, this cell employ sophisticated transport mechanism, and this mechanism can fail, leading to mislocalization of proteins. And aberrant localized proteins can cause human diseases as distinct as Alzheimer's disease, cancer, or kidney stones. So here is just a short list of proteins whose mislocalization can cause diseases in humans. There are two direct therapeutic implications of this. First, you can restore normal protein localization, and you can therapeutically mistarget and thereby inactivate disease-causing proteins. And this latter strategy, a striking example for the latter strategy is periphosin, which is an inhibitor of the oncokinase AKT. And AKT is activated in many cancers, and to be activated, AKT has to be localized at the plasma membrane, and periphosin blocks this localization at the plasma membrane, so mislocalizing AKT and inactivating this oncokinase. So our lab is interested to use the first strategy to restore Foxo protein function. And Foxo proteins are part of the PI3 kinase AKT pathway, which is nowadays considered as the most frequently activated pathway in human cancer. And there are genetic and epigenetic alterations all along the pathway, most prominently within the tumor suppressor gene P10 and the oncokinase PI3 kinase. And Foxo transcription factors actually are very conserved proteins and are the major transcriptional output from this pathway. So Foxo proteins have been established recently as bona fide tumor suppressors. So they block actually many different steps of tumor formation and progression. And since Foxo are transcription factors, they exert their function within the cell nucleus. And in many cancers, Foxo proteins are inactivated via mislocalization to the cytoplasm. So the major inactivating input comes actually from the oncokinase AKT, but there are other oncokinases like ERK or IKK beta also inactivating these tumor suppressors. So the difference between Foxo tumor suppressor and other tumor suppressors like P53 and P10 is that P53 and P10, for example, are mostly inactivated by genetic and epigenetic alterations, whereas Foxo is inactivated mostly by post-translational modifications. So this is a very important difference when it comes to a therapeutic strategy when you want to restore the function of a protein, because in the case of Foxo, the protein is still there and can be reactivated, and that's actually what we want to achieve, to reactivate the tumor suppressor function of Foxo. And in particular, what we want to achieve is to identify targets that can be exploited pharmacologically and small molecules that directly drive Foxo into the nucleus. So we generated a panel of cell-based assays, mostly based on robotic microscopy. What we basically do is we monitor molecular events in disease-relevant context at single-cell level and in large-scale experiments. So we follow these molecular events at different time points or even in time lapse with different concentration, different channels, and we not only follow the molecular event, but we monitor at the same time hundreds of additional parameters. So that's why this technology is called high content screening, because it's high content in information, it's information rich. So actually we discussed the opportunities and challenges of this technology in a recent review, which we entitled High Content Screening, Seeing is Believing. So anyway, with these tools in hand, we performed a massive compound screening. We screened 35,000 compounds. For the first filter was the Foxo translocation, counter screening for nuclear export inhibitors, and then we did a focused kinase profiling. Here just how the images look like. And with this strategy, we ended up with 242 active compounds. This is about 0.7% hit rate. And of these active compounds, 22% were PI3 kinase inhibitors, 6% nuclear export inhibitors, 2% receptor tyrosine kinase inhibitors, and for 70% of the compounds, the molecular target is still elusive, so there's still many things to, it's a gold mine to discover new mode of actions and new compounds. So among the most active compounds against PI3 kinase was a chemical series which is the pyrosolopyrimidines. This turned out to be reversible inhibitors of the PI3 kinase because washout recovered the AKT activity very quickly. And the most active compound was this one with a potency of 400 nanomolar against the recombinant PI3 kinase protein. This is a good starting point, but nowadays in drug discovery, a good lead compound is expected to have low nanomolar activity. So we had to do some optimization, and we used two different strategies: virtual screening and medicinal chemistry to prepare derivates. We prepared 216 derivates, and with this strategy, we ended up with this already very potent compound, which is a IC50 values for the inhibition of PI3 kinase in 20 nanomolar, and in the cell-based assay against Foxo for 40 nanomolar. So it's, as I said, a very potent translocator of Foxo. It's much more potent than the reference compound from Lilly. And so we have a very potent compound, but a very essential point is that this compound would have subselectivity. And here is a typical kinase profiling we do routinely. And here you see more promiscuous compounds inhibiting many kinases, and here more selective ones. And our compound was a very selective one in a panel of 72 kinases. It's a pan-PI3 kinase inhibitor with slight activity also against mTOR. It has activity against a panel of cell lines with genetic alterations within the PI3 kinase AKT pathway, and potently inhibited or induced cell cycle arrest. So this compound potently inhibits the activity of AKT, and which is very important from a therapeutic point of view. It shuts down the whole pathway and didn't modulate unrelated biomarkers. And last but not least, in this business, it's very important to show also the modulation of biomarkers in vivo, and we were able to show this in a transgenic mouse model overexpressing the myristylated form of PI3 kinase alpha, modulating also the biomarkers in vivo. So with a different Repora protein, we were able to identify also novel nuclear export inhibitors. So this is actually a attractive anti-cancer strategy, because many tumor suppressors, not only Foxo, are nuclear proteins which are inactivated by mislocalization in the cytoplasma. So the blocking of its export, or their export, would be a potential anti-cancer strategy. So unfortunately, the paradigmatic inhibitor of the nuclear export is leptomycin B, turned out to be very toxic for humans. Leptomycin B forms a covalent binding with the cysteine residue of an export receptor. So in contrast, our compounds, this is one of our novel nuclear export inhibitors, were reversible ones, holding promise to be less toxic in clinic. However, the difference between a good ligand and a successful drug is that a successful drug is the combination of potency and good physical and chemical properties. And we have actually all the sophisticated tools to analyze these properties, but this part is already beyond the scope of any academic research project. Nevertheless, there are actually an urgent need also for academic research to adopt large-scale biology. So, and I will show you in the next few slides how we successfully used this approach for two independent target discovery projects. So what we first did is we screened a panel of 73 small molecule compounds with known biological actions. This means that we knew what these compounds do. We knew the molecular target of these compounds, and we did this in 3 different concentrations to really hit also the best IC50 values. And we ended up with 18 active compounds driving Foxo into the nucleus. Some of them old known inhibitors of the PI3 kinase AKT signaling pathway, but also some new compounds, and we were actually surprised that 3 different inhibitors of the calcium binding protein calcium calmodulin drove Foxo into the nucleus. So what we first asked was if this effect was specific for this protein, for calcium calmodulin. So we took this compound W12, which is from a chemical point of view almost identical to one of the active compounds, but which with a much less affinity to calcium calmodulin. And here I show you that even high concentrations of this compound were unable to drive Foxo into the nucleus. Then we ask, has this effect anything to do with calcium signaling? So we decreased intra and extracellular calcium with BAPTA and EGTA, and I show you here that this triggered the nuclear accumulation of Foxo. However, when we looked downstream of calcium calmodulin inhibiting all the downstream client proteins of calcium calmodulin, we were unable to reproduce the nuclear translocation effect. However, when we monitored the activity of AKT upon treatment of these active compounds, all of the active compounds had an effect on the activity of AKT. So to make a long story short, we were able to place a new component using our approach, a new component into the already very complicated, very complex signaling network that regulates Foxo. And we were actually quite excited when we learned that calcium calmodulin was also among the priority hits from our RNAi screening. So, but let me explain first how we did this screening. So we used a library of vector-based shRNAs. This library has been prepared, generated in the lab of René Bernards at the NKA in Amsterdam. And this library contained about 24,000 shRNAs, silencing about 8,000 human genes. And we filtered this library with 2 different independent assays. One is dependent on the transcriptional activity of Foxo, and then the translocation of Foxo. And only if the shRNA scored in the 2 independent systems and repeatedly were included into the priority hit list. And here we have, as I told you, calcium calmodulin, also known players in the PI3 kinase AKT pathway, and one gene that drew our attention was TRP2. TRP2 is the mammalian homologue of the Drosophila gene tribbles. It's a pseudokinase, and when we silenced tribbles, Foxo went into the nucleus and triggered Foxo-dependent transcription. On the contrary, when we expressed a V5 fusion protein, the Foxo-dependent transcription dropped dramatically. So since Foxo is an important gene in cancer, we wanted to know if tribbles has anything to do with cancer. So we monitored the expression of tribbles in a cancer profiling array. We scanned samples from normal and tumoral tissues from 154 patients and from 70 different organs. And this is a cDNA array actually. So spotted the normal tissue and the tumoral tissue from a patient side by side. And we hybridized this with TRP2 specific probe. And what called our attention was actually the dramatic overexpression in skin cancer. So 7 out of 10 skin cancer had a really dramatic overexpression of tribbles. So, but when we had a closer look to the case histories, it turned out that 7 of these 10 cases were malignant melanoma, and the overlap with the overexpression was 100%. So when we, and the same was true actually for the panel of cell lines, the cell line with the highest TRP2 expression was also from malignant melanoma. So we were able to confirm this in an independent panel of cell lines. The black bars represent melanoma cell lines, and tribbles. We were able to correlate tribble expression negatively with nuclear Foxo and BIM expression. BIM is a target gene of Foxo, and we were also able to positively correlate TRP2 expression with melanoma stage. So apparently, tribbles is a suppressor of a tumor suppressor. This led us to ask ourselves, tumor suppressor might be an oncogene. So to test this hypothesis in melanoma cell lines, we established a stable knockdown model for TRP2 in melanoma cells. And with these tools in hand, we were able to show that a stable silencing of TRP2 decreases the proliferation, the capacity to form colonies in soft agar, and also to close wounds. And we were also able to show that stable silencing of tribbles inhibits the tumor growth in vivo, in a in vivo xenograft model. So the cells with the tribbles, with the stable silenced tribbles, never formed or almost never formed tumors. In the rare cases when they formed tumors, they never grew beyond a few square millimeters. So I think it's quite clear, but I would like to underline that large-scale biology does not replace hypothesis-driven approaches. So and we were actually able, using the combination of both, in particular an unbiased pathway-based approach, to identify several points of intervention to restore Foxo activity. So, and we're using a candidate gene approach instead of an unbiased pathway approach, we never would have been able to identify these players because these have been associated previously with different signaling networks. So what we have achieved within the PI3 kinase AKT pathway can be adopted for any other signaling pathways. In fact, virtually all the major signaling pathway associated with cancer culminate in a translocation step of downstream signaling molecules. Here are just a few examples. And as I demonstrated, these translocation steps can be monitored using image-based high-content screening at single-cell level in large-scale experiments, and thereby you can monitor the pathway, the signaling pathway as a whole. So this strategy, we think that this strategy, this pathway-based strategy, will be key to future anti-cancer therapy, because the cancer genomes are telling us that cancer is a pathway disease. So the sequencing of cancer genomes actually revealed an overwhelming number of somatic mutations. So obviously the majority of these mutations has nothing to do with tumor formation or progression. But there are also many more driver mutations as expected. Driver mutations really drive the tumorigenesis. This is actually bad news, kind of bad news from a therapeutic point of view, because it became clear that curing cancer just hitting a single molecular target will be the exception. It's possible, like it has been demonstrated in the Gleevec case, but it seems that this will be really the expression. However, many of these driver mutations cluster in well-established cancer pathways. So drugging entire signaling pathways rather than just one genetic alteration at a time will be a more appropriate approach to attack cancer. So I hope I convinced you that our approach, unbiased, image-based pathway approach, is very powerful. But I will tell you that in some cases there is no alternative to use image-based or pathway-based drug discovery. So this is the case, for example, when the molecular target is unknown, or the biochemical assay is not available.